DOE CSGF Fellow Laura Watkins in the field of theoretical chemistry at the University of Chicago.
Hi everyone, thanks for tuning in. My name is Laura and I'm calling from Chicago, where I'm a student in the chemistry department. And today I'd like to talk to you about my research on using computer simulation to study proton transport in the Influenza A M2 channel. So the M2 channel is a critical protein in the influenza virus. It's a bundle of 4 helices as shown here in the center, and one of its main responsibilities is to bring protons in, or hydrogen ions in, from the outside to the inside of the virus. This is important in the infection step of the life cycle. So just a quick overview of what's going on. So you have your virion here, and to get into the cell, it's brought in into an endosome. And to actually infect the cell, it needs to break out of this, and that's done by bringing these hydrogen ions in to make the interior acidic, which allows it to break out and to bring its contents, such as RNA, into the cell so it can replicate and finish the life cycle. And because of this critical function in the infection step, it is a critical drug target. And so 2 of the 4 available antivirals that are available right now target this protein. And so that's amantadine and romantadine is shown here. Unfortunately, there are prevalent mutants of this channel that are all over the place, and these mutants are not inhibited by these drugs. So now essentially these drugs are considered useless because they're ineffective against those mutants. And because of this, that warrants the continued study of this channel to further understand its mechanism, so that new drugs can be designed for these mutants. And so to study this, we're going to use a variant of molecular dynamics. And so just to quickly give an overview of what molecular dynamics is, for those of you that don't know. Molecular dynamics is a atomistic simulation method where you ignore the electronic degrees of freedom of your atoms, and you then take your atoms and you put them together to define molecules, where you define specific set bonds. You can then bring in a bunch of molecules into your simulation box to define your system. And then you then use a force field to define your interaction potential between all of the atoms, which is typically a function of bonds, angles, dihedrals, and non-bonded interactions. And then you use Newton's equations of motion to propagate the system in time and to move it forward by taking small, like femtosecond time steps. For proton transport, however, classical molecular dynamics is not sufficient. So maybe you've seen this equation before from like a general chemistry class, where water dissociates into hydroxide and hydronium. However, when you have this extra proton, hydronium ion does not really exist in water. It's because instead it's part of a complex solvated molecule. And when protons are in water, they don't move as a hydronium ion. Instead, they hop between water molecules through this known as the Grothaus shuttling mechanism. And therefore, classical molecular dynamics, where you have fixed bonds, is not sufficient to study this. And so instead, we use what we call multiscale reactive molecular dynamics, which is a method specifically that my group has developed. And a very kind of high-level overview of how this method works. You can imagine that at each time step, you take your system and you redraw all of the possible ways that the bonds could be arranged. So for example, in this state 1, that excess proton is on the center water molecule. You could also imagine redrawing the bonds such that this excess proton is on the top water molecule. So this algorithm does, in every time step, it finds all of the possible states like this with different bonding topologies, and then takes a linear combination of them to best describe where the proton is, and we call that the center of excess charge position. And so by doing this, we're able to explicitly model the reactive proton, and we're able to capture behavior like this proton hopping mechanism shown here. So I was able to take this method and apply it to the M2 channel and ask the question of how can we better understand the proton transport mechanism by looking at these specific interactions between the proton, water, and the channel. And so here is just a first kind of qualitative look of the kind of things that I looked at, where you can look at, as the proton moves through the channel, how do the waters respond and what's going on. And so you can see here, if you look at it a little bit closely, you can start to just see differences in the structure of the water. And so, of course, we can quantify this. So here, by plotting the direction of hydrogen bonds between water molecules with respect to the channel axis, and here we're plotting with respect to the proton position, or the center of excess charge position, as the proton moves through the channel. And so it's clear to see just by the trends that the directions of hydrogen bonds essentially flip directions when the proton is in the region of this water. You can see that as well in this picture, where when the proton is outside of the channel, hydrogen bonds are primarily pointed up, versus when the proton is right there directly above this region of water, the hydrogen bonds flip directions, and the waters are able to flexibly and dynamically reorient to allow the proton to pass through. And another thing that I studied in this project is the proton's path through the channel and how that relates to the asymmetry of the channel. And so here on the left, you can see a density of where the proton likes to be as it travels through the channel. And then on the right is a slice of this density, where we're looking just at a slice through the channel and looking at a 2D histogram of where the proton likes to be. And so it's clear that on this nanosecond time scale of simulation, one, the channel is not symmetric, and two, that the proton has specific hotspots that it likes to stay in. And so this is important, one, for drug design. This is an important feature that in considering how to design drugs, that you could take advantage of these features. And together, both of these results and other results really show how static structures from crystallography or other experimental methods don't show the full story of proton transport. And it's really difficult or impossible to understand how proton transport works without looking at this dynamic picture. So then I took this a step further and looked at what we can learn about drug binding by looking at the proton transport mechanism. So this is a crystal structure of amantadine in M2, and this is one of the inhibitors that we have available for wild type M2. And amantadine is a charged species, just like the proton. And so my question is, what can we learn about looking at the proton mechanism and how the channel stabilizes and allows the proton to move through? How does that relate to where the drug binds and why these are effective inhibitors? And so this is a plot of the hydrogen bond occupancy of the alanine 30 carbonyl with water in the channel. You can see it's pretty stable as the proton is outside of the channel. But once the proton in this region approaches the ala 30 carbonyl, the occupancy significantly decreases. And what this shows is that these flexible interactions are able to help stabilize the excess charge. So in a sense, the ala 30 carbonyls are sharing their waters with the proton as it's in this region. And this is also the region right where the charged group of the drug binds. And so this is likely indicating that these same flexible interactions are able to help stabilize the drug. Another aspect I looked at was the shape of the pore and the dynamics of that shape during proton transport. So this is essentially showing that at the top of the channel where these two residues are, it's pretty stable, pretty circular. And whereas at the bottom of the channel where these three residues are, as the proton moves through the channel, it's less circular, and there's also much more change. And so the drug binds up in this upper portion of the channel where these two residues are. And since the drug is a relatively spherical shape itself, this is likely a critical feature as well of the channel that enables this efficient drug binding. So that's just a very brief overview of some of the results and some of the things that I've done. Altogether, these results show that proton transport is incredibly dynamic. There are many people that try to look just at static structures and look at water wires to determine whether proton transport is possible or whether it exists in a new system. And what I've shown here and what other people in my group have shown is that proton is actually frequently able to pave its own path, in a sense, where even if there aren't waters where it needs to go, it can draw water in, and it can dynamically interact with the water and the protein and its environment to get where it needs to go. And so these are just a couple other results that I have worked on, and another project that I'm still finishing up. So all of this, you might say, wow, this looks a lot like biology. Or does this relate to COVID-19 at all? It's a great question. And so this is a published structure that just came out last week as of recording this. And this is the protein P3 from SARS-CoV-2. And this protein is shown to have some ion transport function, although there's still many questions about it, about its function, and even about its structure and whether this is the full story here or not. But I'm hoping to use a similar approach to what I just shared with this protein, and hopefully work out some of its mechanism and how it works to be able to inform future drug design. So with that, I'd like to thank the DOE and the Krell Institute for providing this fellowship and for the opportunities that it's provided for me and the support. And of course, I'd like to acknowledge my advisor, Greg Voth, and our collaborator, William DeGrotto and UCSF. And I'd also like to thank all of my fellow fellows, and sad I don't get to see you guys this year, and I hope you're all doing well. Thanks.